Hello, thank you for inviting me to talk today. I am Stephanie Piecewicz and I'm representing Bell Biosystems, and I'm going to talk about synthetic organelles for imaging and other advanced applications in cells. So this is a little bit about our company. We have a seasoned management team. We're located in San Francisco. We have a little bit of money raised and we've been engaged in R&D for a little over 2 years. We continue to expand our patent portfolio, and I'm going to talk about our Magnelle product, which will be in beta preclinical release at the end of this year. So I'm constantly impressed by the work that's presented at this meeting toward the development of cell therapies, but clearly there's a backlog in the translation of these therapies to patients. And if you just focus on the green bars, which represent early preclinical work and early clinical phase trials versus the blue, which are late stage clinical trials, you can see that there's a large gap, especially in such fields as cardiovascular disease and CNS disease. And this has implications into products that are approved in those spaces. So the development of tools that can help us understand the function of those cells in vivo can help bridge that gap to translation. So what are some of the features of tools that can help us understand the function of cells in vivo? One, the ability to better and more efficiently steer cells to sites of interest. Two, the ability to image cells over time in vivo. Three, the ability to ablate and destroy cells if they function undesirably in the body. And also the ability to control cells in vivo. So our solution is to develop a tool that will have all of these features. We're engineering a naturally occurring magnetic bacteria to live within cells as a magnetic organelle, which is what we call a Magnelle. And this will provide the cells with a magnetic phenotype that will serve as a living MRI contrast reagent for imaging. And also we can use an alternating magnetic field to destroy the cells using magnetic-induced hyperthermia. And also the genome of the bacteria can be taken advantage of to express genes and proteins of interest into host cells. So this is what the Magnelle looks like when it's transfected into a cell. And I want to point out that we have some preliminary acute toxicity studies that showed positive results. And also we see no significant changes in basic cell function in the cell types that we've tested. So right now we're focusing on developing our preclinical reagent and focused on developing that for imaging applications. So this is what the preclinical workflow looks like. So cells are labeled ex vivo. We've developed a magnetofection method where cell culture plates are placed directly on a specially designed magnetic plate, and by physical means the Magnelles are taken up into cells, and then those magnetic cells are transplanted into an animal and imaged using well-established MRI methods. So we have successfully labeled many different cell types, and these are some representative examples of those cell types that we've labeled. Magnelles are shown in the bottom panel in red. And these are representative examples of stem cells, T cells, and cancer cells that we've labeled. And you can see very high loading of Magnelles in those cells. We get on the order of 5 to 20 picograms of iron per cell. These are representative images of Magnelle-labeled cells in vivo. On the left are Magnelle-labeled cells injected into the liver. And on the right are Magnelle-labeled cells injected into flank muscles. So in addition to a preclinical reagent, we'd also like to develop this for use in humans. Currently, cells are prepped ex vivo and transplanted into humans, and commonly there's no tracking or imaging of those cells once they're transplanted. So we envision Magnelles being incorporated into cells prior to transplantation of these cells, and this would enable monitoring using MRI and also the option to destroy those cells using clinically approved magnetic hyperthermia equipment. So in summary, Magnelles are a tool that can accelerate the development of cell therapies. Magnetic cells and MRI can provide translational data and safety features, And this living contrast agent can enable longitudinal studies of cells in vivo. No genetic modification is required to the host cell, so this minimizes perturbation to the cell itself. And there's also a huge genetic capacity in the bacteria, so we can envision interesting multimodal and functional capabilities of those cells. So we have a couple of opportunities for partnering, both in the preclinical business and clinical business. We're looking for customers to try our prototype and also some commercialization partners. And also we're actively seeking clinical co-development partners, and we see high value in the cancer immunotherapy, CNS, and cardiac disease space. So thank you, and I'm happy to take any questions if there's time for that. Okay. Yeah.
In your mouse images, what was the field strength of your mouse?
7 Tesla. I believe 7. But we've done anywhere from 1 to 11.7. And that will affect sensitivity and all that. So. Oops. Yeah. So we've been able to detect a single cell in vitro using a very high strength magnet from one of our collaborators. But yeah, you would need a very high strength magnet to do that. Okay, thank you.